Hello everyone, I am A. Sabina Banu, MSc Microbiology, Sacred Heart College, Autonomous Tirupattur. I like to present my topic single cell protein used as food and feed. The concept of this topic a new way to produce protein using microorganisms which is helpful for both humans and animals in agriculture. Now I started from the history of single cell protein post World War II, late 90s, 1940s to 1950s, origin of the concept. After World War II, the global population began to grow rapidly, so this led to increased demand for food and protein, especially in developing countries. So traditional protein sources like meat, soy and fish were insufficient to meet demand. Scientists began exploring non traditional microbial sources of protein to address global protein deficiency. In 1960s, introduction of the term is single cell protein. The term single cell protein was first introduced by Carol L. Wilson, a scientist at the Massachusetts Institute of Technology in the 1960s. It referred to dried microbial cells or protein extracted from microorganisms like bacteria, yeast, algae, fungi. These proteins were originally aimed at being animal feed supplements but later explored for human consumption as well. In 1970s, industrial production and commercial interest. In 1970s, industrial production of single cell protein began during this decade, especially in countries like the UK, USA and Germany. So the companies like Empirical Chemical Industries developed large scale single cell protein projects. Example ICI used the ICI used Methylophilus methylotrophus, a bacterium, to produce single cell protein using methanol. Single cell protein was also produced using substrates like agricultural waste like straw, molasses, then petrochemical byproducts, methane, methanol. However, high production cost and safety. In 1980s, focus shifted to safer and sustainable single cell protein. Researchers shifted their focus from bacteria grown on petrochemicals to more natural and safer sources like algae, Spirulina chlorella, fungi, Fusarium venatum used in corn, yeast, Saccharomyces cerevisiae. Single cell protein became popular in the health food industry and vegetarian protein alternatives. 1990s to present, technological advancements and new applications. Biotechnology advancements led to better fermentation techniques, improved strain selection, safer and more efficient single cell protein production. Then single cell protein found a broader uses in aquaculture feeds as a replacement for fish meal, poultry and livestock feed, human health supplements like spirulina powder. In two cases today, global recognition and research, single cell protein is now being recognized as a sustainable and eco-friendly protein alternative. It is being integrated into meat substitutes, protein bars and powders, pet food and aquaculture diets. Research is focused on using wastewater and agro waste as a growth medium, genetic modification of microbes for higher yields, ensuring food safety and public acceptance. So now let's see introduction to single cell protein. Single cell protein refers to microbial biomass used as protein source for food and feed applications. Single cell protein is produced from various microorganisms such as bacteria, yeast, algae and fungi. It offers a sustainable alternative to traditional protein sources in agriculture. Types of microorganisms used in single cell protein production. Bacteria, yeast, filamentous fungi, and algae are commonly utilized for single cell protein manufacturing. Each microorganism type offer unique nutritional profiles suited for different applications. The choice depends on factors like growth rate, ease of cultivation, and protein content. Single cell protein extracted from algae, fungi, bacteria, and yeast, both animals and humans. Industrial production of single cell protein. So, first, selection of microorganisms. Microorganisms should grow fast and give high protein yield. Commonly used microbes: algae, yeast, fungi, bacteria. In under the algae, spirulina and chlorella; yeast, Saccharomyces cerevisiae; fungi, Fusarium venatum; bacteria, Methylophilus methylotrophus. The organism must be non-toxic and safe. So, selection of substrate. Cheap and easily available materials are used to reduce cost. Examples: molasses, agricultural waste, food processing waste, methanol or methane. Using waste helps in waste management. Culture medium preparation. Nutrients like carbon, nitrogen, minerals and vitamins are added. The medium should support maximum microbial growth. Then fermentation process. Single cell protein is produced using fermentation technology. The two main types are submerged fermentation and solid state fermentation. Fermentation is carried out in large bioreactors. Control of growth conditions. Important parameters like temperature, pH, oxygen supply, agitation. Proper control ensures high yield and quality single cell protein. Then harvesting of biomass. Microbial cells are separated from the culture medium by filtration or centrifugation. This step collects the protein rich biomass. Then downstream processing includes washing, drying, grinding into powder. Nucleic acids are reduced to make single cell protein safe for human use. Then quality and safety control. Single cell protein must be free from toxins, free from harmful microbes. It should meet food and feed safety standards. Then packaging and storage. Dried single cell protein is packed as powder, tablets, feed additives, or proper storage increases shelf life. Then economic and environmental benefits. Needs less land and water than traditional protein sources. Converts waste into useful protein. Environment friendly and sustainable. The nutritional composition of single cell protein. Single cell protein is rich in high quality proteins, amino acids, vitamins, and minerals. The protein content generally ranges from 50% to 70% of the biomass. It can be modified to improve particular nutritional properties for various applications. Examples of single cell protein products available in the market. So, first one, single cell protein source like fungi, Fusarium venatum. Example product name Quan. Description and use: microprotein based meat alternative, chunks, nuggets, sausages used for human food. Then algae, spirulina powder, rich in protein, vitamins and antioxidant, health supplements. Yeast, Saccharomyces cerevisiae, pro yeast, feed yeast. Description and use: dried yeast protein added to poultry and pig feed, used for animal feed. Sources of single cell protein. Microalgae such as spirulina and chlorella are cultivated for their high protein content and health benefits. They can be grown in diverse environments, including wastewater, making them environmentally friendly. Then yeast. Yeast like Saccharomyces cerevisiae are used in fermentation process and as single cell protein in animal feeds. They are rich in protein and can utilize various substrates including agricultural waste for growth. Then fungi. Filamentous fungi such as Fusarium venatum are utilized to produce mycoprotein, a popular meat substitute. They grow rapidly and can convert inexpensive substrates into high protein biomass. Then bacteria. Certain bacteria can produce single cell protein through fermentation. They are efficient at converting organic waste into protein, contributing to waste management and protein production simultaneously. Production methods for single cell protein, like already we see: fermentation technology, bioreactor system, substrate utilization. So fermentation technology. Single cell protein production often involves fermentation, where microorganisms convert sugars or other substrates into biomass. This method is efficient and scalable, suitable for industrial application. Then bioreactor system. Controlled bioreactor systems optimize growth conditions for microorganisms, ensuring high yields of single cell protein. These systems can be designed for various microorganisms, enhancing the efficiency of protein production. Then substrate utilization. Various substrates can be used for single cell protein production, including agricultural residues, food waste, and industrial byproducts. This provides a cost-effective protein source and reduces waste. Applications in animal feed, livestock nutrition, aquaculture, pet food. Microorganisms and examples of microorganism, type of protein, protein content, and used for. So now such as microorganism like algae, yeast, fungi, bacteria. Algae example organisms Spirulina platensis. Type of protein is present, algal protein. Protein content, it contains 60 to 70% used for fish feed, poultry, pet food. Applications of human food from single cell protein, meat alternatives, protein supplements, nutraceuticals. Then advantages of using single cell protein in agriculture. Single cell protein production is environmentally friendly with lower land and water requirements. It provides a high quality protein source that can utilize waste streams. Single cell protein production can contribute to food security and reduce ecological footprints. Challenges and considerations in single cell protein adoption. First one, consumer acceptance. Education and awareness campaigns are essential to inform the public about its benefits and safety. Regulatory challenges. Ensuring compliance with food safety standards or regulations is critical for market entry. Then technological improvements. Continued research and development are needed to enhance production efficiency and reduce cost. Future prospects of single cell protein. Innovation in production, like advances in biotechnology and fermentation technologies, are expected to enhance single cell protein production, making it more efficient and cost effective. Then integration into food systems. Single cell protein is likely to be integrated into various food systems, providing a sustainable solution to global protein demands, particularly in developing countries. Research and development. So this single cell protein having also disadvantages, like high nucleic acids may cause health problems like gut if not removed. Hard to digest. Some single cell protein like algae is not easily digested. Bad taste or smell. Some single cell protein has an unpleasant flavor. Risk of contamination. Harmful microbes may grow if not handled properly. Then allergy. Some people may get allergic reactions. Not accepted by all. People may not like eating protein from microbes. Costly processing. Needs extra steps to make it safe and tasty. Toxins. Some microbes can produce harmful toxins. So previous CSIR net questions asked asked from the single cell protein topic. In 2019, they are asking which statements are true about single cell protein. So the correct answer is A, C and D. Like single cell protein includes protein from bacteria, yeast, fungi and algae. Then single cell protein is rich in protein, vitamins and minerals also. Then single cell protein can grow on waste like molasses or agricultural residues. In 2020, which of the following are challenges in human single cell protein consumption? The correct answer is A, C, D. Like high nucleic acid content, risk of contamination, regulatory and public acceptance issues. In 2022, select microbes commonly used for single cell protein. The answer is Methylophilus methylotrophus, Saccharomyces cerevisiae, Spirulina platensis. Then in 2023, advantages of single cell protein in agriculture. The answer is high protein yield per area, uses agro-industrial waste, then reduces environmental pollution. In 2024, according to Unacademy Integrated Study, single cell protein refers to production of fermentation of waste products. Thank you.